Hey guys, so I'm the SI leader for Biology 101, and we talked about mitosis last lecture. So let's go on ahead, get started. We're going to do a brain dump activity first. So we're going to need a scribe to go up and be silent and just write down what everyone else has to say of what we remember from the lecture about mitosis. So who is willing to be the silent scribe?
I will.
Awesome, thank you. All right, so now we need to give our awesome silent scribe everything we remember about mitosis. So this could be the names of the phases and then like some slight descriptors about what happened, where they happen relative to each other, stuff like that.
Isn't it interphase where it grows?
Good, all right.
I remember the professor talking about metaphase.
And then he said something about like cytokinesis where like they pull apart into two cells.
Going along with that, I think anaphase is where they just started to pull away.
What started to pull away there?
The cells, or
Can we help
I
her
think
out?
it's the chromosomes.
Yeah,
pull
she's
away?
right. Yeah, I think that's right.
Yeah.
okay, that sounds right.
Because then I think it's telophase when the cells start to pull apart, right?
That sounds right, but
Yeah.
I don't remember exactly.
Mm-hmm. Yeah.
Is that all of them?
I don't know. Are we all in agreement that this is the complete list?
I think we're missing something.
We are missing something. So what are we missing?
It's prophase.
Prophase. Good.
Yeah.
All right. So now a good thing to do would be put these into chronological order. So let's just start at the top. Interphase. Where does that happen in relation to everything else?
First.
First, yeah. So that's just where the cell grows. It's just before any division happens. Good. So now after interphase, what happens?
I think prophase.
Good. All right. And then after prophase?
Metaphase?
Good. And do we have a descriptor for metaphase? Like what's just in general happening in metaphase? Something
I remember
iconic.
like M is kind of like for middle. Like, that's when the chromosomes align in the middle.
Mm-hmm. Yeah. That's a pretty iconic thing for metaphase. And then prophase, do we have anything noteworthy there? Just some brief descriptor?
Isn't that when all the chromosomes start to form instead of just being like a giant ball of yarn in the middle?
Yeah, good. The chromosomes begin to take shape during prophase. That's awesome. All right, so then we have interphase first, prophase next, then metaphase. What happens after metaphase?
I don't know, but I know cytokinesis is at the end.
All right, so we have 6 of those. So we can just go ahead and put a 6 there, yeah. All right, so we have 2 left, anaphase and telophase.
Anaphase is before telophase.
All right.
Because if it attaches to the chromosomes, it has to do that before the cell splits.
All right, yeah, that's good logical thinking. Good. All right, so we have all the phases for mitosis, and we have them all in order. So now we're going to split up into pairs. Thank you, Scribe. Thank you for your help. So now we're going to split up. We have 1, 2, 3, 4, 5, 6. 6 people here. So we'll split up into pairs. Each pair will have 2 of the phases. So let's start off by counting off to 3.
1.
2.
3.
1,
2, 3.
Awesome. So we have 3 pairs. Let's have the first pair here. You'll have the first 2 phases. We'll have pair 2 here. You'll have the next 2 phases, and then pair 3 on this board for the last 2 phases.
All right.
So now we're just going to go through and draw what the cell looks like. So just a little pictorial diagram of what happens in the cell during each of the phases. So just what the cell looks like. Be sure to focus on the nucleus. And then if you have any expansion to the description that happens that we have from the brain dump, go on ahead and add that with your drawings.
I think we have interface now pretty well. Just the yarn. With the,
Yep.
how are you feeling about prophase? I'm kind of not sure what happens at this step.
Well, because I think at this step, ball of yarn is starting to form into chromosomes, but I don't know what's going on with the nucleus. So if we just draw it like this for now, I don't know what else besides that. I think we should ask Nathan.
Yeah, I don't know. Hey Nathan, we're kind of lost on the prophase step of what goes on with the nucleus and.
Because we know it's starting to like become chromosomes, but we don't know how to draw it.
All right. So a good place to start might be asking a fellow student. Maybe someone from another group will remember how to do prophase, and maybe they can give you a hand.
Okay.
Okay. Hey, do you guys know what goes on in the prophase step for the nucleus and the chromosomes? We're kind of lost.
I think I remember. I can probably help out.
What do you guys have so far?
Because we got the nucleus still, and we know that the chromatids are starting to become chromosomes, but we don't really know how to draw it.
I think they kind of look like this is a good way to represent them.
Okay.
Then I also think this is when the envelope starts to break down. So you probably just like do that to show.
Oh, that's right.
Does that sound good with you guys?
Yeah, looks
yeah,
good.
yeah, that makes a lot
All
of
right,
sense.
glad
for
to
it.
help.
Thank you.
Thanks.
Okay, yeah, that makes a lot more sense. That's
Yeah.
where they start forming and then the nucleus breaks down.
Yeah, and then I think these things are still just floating around, so just have those there.
I'm happy with that.
I think we got it.
Did we label the parts?
Yeah, that would be a good idea.
Okay. So these are the chromosomes.
And I think I remember that these are the centrioles.
Oh, okay, yeah. Do you remember what those are called?
No, I don't.
I don't either. Maybe someone else does.
Yeah.
Let's ask them.
Hey guys, do you remember what these are called?
No. No. I guess we should ask.
Yeah.
Hey, Nathaniel.
Yeah.
We're a little lost.
Okay.
We're labeling the parts, but we can't remember what this is called.
Do you remember where in the lecture he was talking about that, the professor?
In the beginning?
I think in the beginning.
Towards the
Yeah.
beginning? All right. Do you guys have your notes with you? Because it might be there towards the
I
beginning
did
of the notes
bring
for
mine.
today.
Let's see. I guess I can flip back through. Oh, here he's talking about metaphase.
All right. Good. Yeah.
Oh, microtubules.
Oh, that sounds right.
Yeah. Good. Awesome. Yeah. It looks like it's coming along well.
Yeah. Thanks for your help.
You're welcome. All right, it looks like we're all done with our drawings and descriptions. So now we're just going to go through chronologically all the steps of mitosis, and each of you will just explain your group's drawings and briefly just what happens in that phase. So let's start over here with interphase.
So with interphase, cells kind of just hanging around. It's still got the nuclear envelope, and the chromosomes are kind of just bunched around. And then prophase, that nuclear envelope starts to disintegrate, and those chromosomes start to form.
All right, good. And now we go from prophase over to metaphase.
So what we have for metaphase, and then we just said that the chromosomes align in the center of the cell along the metaphase plate, and we have all the parts labeled. And then
In,
go to anaphase.
yeah, in anaphase, we have that the chromosomes begin to pull apart and they move to the opposite ends of the cell.
All right, good. And then we go from anaphase over to telophase.
So in telophase, we have that like it starts to cleave in the center, and that's why we have the line there. And then also the nuclear membrane starts to reform after it was broken down.
And in cytokinesis, it splits into two identical cells, and the nuclear envelope reforms.
All right, good. So in cytokinesis, I see you have written down that two identical cells form. is there a specific name for those two identical cells?
It's daughter cells, right?
Yeah. Yeah, daughter cell is good. That typically comes up in the exam, so that's a good thing to have remember in your memory. And then one more thing about this. So we started at interphase, and we had wonderful explanations and drawings all the way to cytokinesis, but then we end up with two identical daughter cells. What happens after that? What happens to the daughter cells?
What do you mean by that?
So we went through mitosis. We began over here with interphase. So then the professor mentioned that this process happens multiple times in your lifetime, just some really large number. So then how is that large number sustained throughout your lifetime with these daughter cells? So the daughter cells are half the size of the original cell. And then after this, they have to go on to do something else. So where do they go after this?
I think they go back to interphase, and then the whole cycle starts over again.
Yeah, good, because remember at the beginning we said interphase is where the cell grows, so since the daughter cells are half the size, they'll grow back up to be mature cells to go on to prophase later on.
Okay.
So good
that
work.
Makes sense.
All right, before we go, we're going to do a closer activity. It's going to be a clear and muddy chart. It's a good way to see what you know and what you don't know, and we're going to do this with our peers, so maybe your peers will think of something that you didn't think of. So I'm just going to put up here on the board a clear column and a muddy column, and then within your partners here, just discuss what you think you got out of this activity, what you feel like you have a solid foundation on now. We'll put that in the clear chart, and then anything that's still unresolved from the session we'll put in the muddy chart, and then just one person from each partner come up, write down what you came up with for the clear and the muddy side, and then you're good to go. My next session is on Thursday at 6:30 p.m. over in Walk. So that'll be going over meiosis, so what Professor Green went over in the next lecture. So going ahead and discuss clear and muddy.
I have no idea.
All right, good. So let's go through the clears and the muddies that we have. So it looks like for the clears, we have two of the same. Daughter cells are identical. That's good. That just means that it's extra clear. So does anyone have any questions about daughter cells being identical? Anything that's confusing about that? Any aspects at all?
I guess what size are the daughter cells?
What size is the daughter cells? That's a good question. Does anyone remember what the professor mentioned, or maybe the people who had cytokinesis? Maybe they are a good place, Jacob?
I know that they're smaller than the original cell.
Mm-hmm. Yeah.
Wasn't it was it half the size exactly?
Is it half the size?
I think so.
Yeah.
Yeah,
that
I agree
sounds
with you.
right to me.
Because it
Okay.
splits down the middle. Yeah.
Yeah.
Yeah, so
Okay.
that would make sense.
Gotcha.
Good. All right, and then cell growth equals interphase. So cell growth happens during interphase. Anything muddy about cell growth happening in interphase?
Yeah, one thing. So the professor said that cells don't grow, they divide. So I'm confused what you mean by cell growth.
Oh, that's a really good thinker question. So, cell growth. Who had interphase? Who explained that to the group?
So the thing we were saying about that is because they split it into two cells that are half the size of the original,
Yeah.
they have to sort of make that size back up because otherwise your cells would just keep getting smaller and smaller.
Okay.
So they're just getting back to like their original size, and that's what we meant by growth.
Okay.
Aren't like they developing all the other parts of the cell too?
Yeah.
Phase. Okay.
All right, good. Any other questions about the clear side? Okay, good. So now we have the muddy side. Protein that moves the chromosomes. Does anyone know what protein moves the chromosomes?
I definitely didn't remember that.
Aren't there motor proteins that do that job?
Mm-hmm. Yeah. Does that sound good to everyone else?
Oh, now that you mentioned that, I actually do remember.
Yeah.
I think in my notes I had that written down.
Makes more sense.
All right, good. And then nuclear envelope breakdown. So when does the nuclear envelope break down?
I
This
know we
happened in one of the stages.
I know we talked about it, but I couldn't remember because there were like 2 membranes that no one, which was breaking down.
Yeah, that is understandable.
I think it's prophase because we drew it like that earlier.
All right, good. So who had prophase?
We did.
Mm-hmm. Do you guys? Does the nuclear envelope break down during prophase?
It does.
All right, good. So microtubules in metaphase.
So yeah, I was just kind of confused because I know that they're supposed to help pull the chromosomes, but I don't know like what they grab onto to get them to pull apart or how that works.
All right, so it sounds like the question is, where do microtubules grab onto during metaphase? What happens during metaphase with the microtubules? Does anyone know this? Who had metaphase?
That was us. Do you remember?
Are there centromeres at the center?
Center of what?
The chromosome.
Oh, at the center of the chromosome. Yeah.
Oh, that's right.
Yeah.
Does that sound good to
That
everyone?
sounds right.
Yeah.
Yeah. All right, good. So any other questions about the muddy?
Not for me.
Nope.
Any
I'm
other
all good.
questions at all about mitosis?
I
Nope.
think we're good.
All right, I think we're out of time. So thanks for coming, you guys. Reminder, next session, Thursday, 6:30 in Walk. We'll be going over what Professor Green went over about meiosis. So thanks for coming. Hope to see you guys
Awesome.
again.
Thank
Thanks.
you.
Very much.
Gotta go study now.